So friends, I welcome you to my YouTube studio of Theriogenology, and in a series of lecture we are discussing the various topics on Theriogenology. And in our today's lecture we will be talking about the spermatogenesis in the male. Friends, I am Professor GN Purohit, the Head Department of Veterinary Gynecology and Obstetrics, College of Veterinary and Animal Sciences, Bikaner, Rajasthan, India. And in our today's topic, we will be talking about spermatogenesis. The topic 1 will be spermatogenesis, topic 2 spermatocytogenesis, spermiogenesis and spermiation, topic 3 spermatozoa in different species, topic 4 factors affecting spermatogenesis. So we start with spermatogenesis. The male reproductive system has often been referred to as a manufacturing plant, largely because of the continuous production of the fertile spermatozoa. The spermatogenesis is a finely regulated process of germ cell multiplication and differentiation, leading to the production of spermatozoa in the seminiferous tubules of the testes. The germ cells of the seminiferous tubules undergo a series of cellular division and cellular changes. The spermatogenesis has been divided into 2 or 3 stages. Some consider 1 and 2 together. The first one is spermatocytogenesis, which involves mitosis, and mitosis is species dependent. Then the second one is meiosis, and the third one is spermiogenesis. So we discuss each one of them. Spermatocytogenesis is the proliferative stage in which primitive germ cells, the spermatogonia, are multiplied by a series of mitotic divisions to produce the primary spermatocytes. The primary spermatocytes then undergo meiotic divisions, which produce the haploid uh, stage, the spermatid. The primary spermatocyte divides meiotically, meiosis 1, into 2 secondary spermatocytes, and each secondary spermatocyte then divides into 2 equal haploid spermatids by meiosis 2, and the division of the secondary spermatocyte into spermatids is known as spermatidogenesis. Spermiogenesis is the differentiative phase in which the nucleus and cytoplasm of spermatid undergo morphologic changes to form the sperm cells. Metamorphosis of spermatid into spermatozoa is called spermiogenesis. Now, this is the typical sequence of the spermatogenesis. The spermatogonia are divided into A1 and then B1. Then this involves mitosis. This is the spermatocytogenesis. The primary spermatocytes are formed, and then the primary spermatocytes divide to form the secondary spermatocytes, and the secondary spermatocytes produce the round spermatid, and the round spermatid then go morphologic changes to which through spermiogenesis to form the spermatozoa, and the release of spermatozoa into the testes is known as spermiation. So this is how the spermatocytogenesis occurs. Spermatogonia are activated by a number of factors, some of which are still unknown, and then they produce the spermatogonia type A. The spermatogonia type A further divide to form the reserve pool of the spermatogonia type A and the spermatogonia type A1, which then divide to form the spermatogonia type A2, and then the type A3, A4, and then the intermediate spermatogonia and spermatogonia type B. The spermatogonia type B divide to form the primary spermatocytes. 5 mitotic divisions produce 16 spermatocytes. Then meiosis, the primary spermatocytes are transformed to spermatids, which is known as spermatidogenesis. The primary spermatocytes have 46 chromosomes. The secondary spermatocytes have half the number. The first meiotic division takes place, and the second meiotic divisions take place to produce the spermatids, which have the 23 chromosomes. So this is again a picture to depict The spermatogonia are converted to primary spermatocyte in 15 days, primary spermatocyte to secondary spermatocytes in 15 days, secondary spermatocytes to spermatids in 1 to 2 days, and spermatids to spermatozoa in 15 days. Then spermatozoa, they undergo epididymal maturation in 21 days. Then spermiogenesis or spermatoleiosis is the morphological conversion of round spermatid into spermatozoa without a division. The major changes that occur during spermatozoa include the nuclear condensation, the thickening and reduction of the nuclear size, condensation of the nuclear contents into the smallest space. Then there is formation of the acrosome, a cap containing the enzymes that play an important role in the penetration through the zona pellucida of the oocyte. Then there is flagellum formation and generation of the sperm tail. Then there is cytoplasmic reduction, elimination of all unnecessary cytoplasm. Spermiogenesis involves 4 phases: the Golgi phase, the cap phase, the acrosomal phase, and the maturational phase. The Golgi, they divide to form one centriole gives rise to flagellum, other gives rise to the basal plate. The nucleus moves to the periphery, you can see here, the nucleus moves to the periphery, and uh, chromatin condenses. The spermatid rotates, so tail sticks out of the lumen. Here you can see this tail, and then mitochondria congregate around the flagellum and become localized in the midpiece. Most of the cytoplasmic mass is engulfed by the Sertoli cells when spermatozoa are released into the seminiferous tubules, into the lumen of seminiferous tubules. Small amount remains at neck, which as the proximal cytoplasmic droplet. The proximal droplet moves to distal end of the middle piece during epididymal passage. Distal droplet is lost during storage in the epididymis. Now there are the Golgi phase. During the Golgi phase, the newly formed spermatid has a well developed Golgi apparatus. You can see here the small vesicles of the Golgi fuse to larger secretory granules called proacrosomic granules. Here are the proacrosomic granules. The vesicle fusion continues until a large acrosomic vesicle is formed that contains a dense acrosomic granule. This is the acrosomic granule. Then during the last half of the Golgi phase, the centrioles migrate to a position opposite to the acrosomic vesicle. Centrioles, you can see, are migrating. The proximal centriole, the PC, gives rise to the attachment point for the tail, and the distal centriole gives rise to the developing exoneme inside the cytoplasm of the spermatid. The exoneme is the central portion of the flagellum. So during the Golgi phase, there is development of the acrosome and migration of the centrioles. During the cap phase, the acrosomic vesicle flattens and begins to form a distinct cap consisting of an outer acrosomal membrane and inner acrosomal membrane. The Golgi migrates, you see Golgi migrates towards the caudal part of the cell. The acrosome development continues and the flagellum elongates. The flagellum, it is elongating. Then there is the acrosomal phase. Spermatid nucleus begins to elongate. This is the nucleus, it's going to elongate. Acrosome covers the majority of the anterior nucleus. You can see here the acrosome forms the majority of a unique system of microtubules known as the manchet extends from the posterior nucleus. Portions of this one, you can see here, this is the manchet. Portions of manchet attach to the region of the nucleus just posterior to the acrosome. Some of the microtubules of the manchet will become the post-nuclear cap. During the acrosome phase, spermatid become deeply embedded in Sertoli cells with their tails protruding towards the lumen of the seminiferous tubule. The acrosome development continues and the flagellum elongates. The manchet is a series of microtubules associated with a nuclear elongation. Then there is the maturation phase. The manchet migrate towards the tail and begin to disappear. Mitochondria are assembled. Here you can see the mitochondria are going to assemble around flagellum to form the middle piece. Dense outer fibers form around the flagellum. This is the flagellum. Annulus forms the juncture between the middle piece and the principal piece. During later stages of spermiogenesis, Sertoli cells forms the cytoplasm remaining after engulfs the cytoplasm remaining after elongation of spermatid into a spheroidal lobule called residual body. The formation of residual bodies completes the final maturation and spermatids are ready for release. Sertoli cells also remove the degenerating germ cells. Now we talk about spermiation. Spermiation is the release of mature elongated spermatozoa into lumen of the seminiferous tubules. The elongated spermatids, which are oriented perpendicularly to the tubule wall, are gradually extruded into the lumen of the tubule. You can see here, gradually they are released into the lumen. They will have a cytoplasmic droplet, distal cytoplasmic droplet. So this is how actually this is the Leydig cells. These are the interstitial space. These are the tight junctions, Sertoli cells, and these are the elongated spermatids and spermatozoa. The sperm maturation. We talk about sperm maturation. The sperm maturation occurs in the epididymis, which is the place for storage and maturation of the sperm. In the caput of the epididymis, there is fluid reabsorption, sperm chromatin compaction. In the corpus, there is protein synthesis, vesicle secretion, acquisition of sperm progressive motility, and sperm chromatin compaction. In the chorda is the site of epididymis for sperm storage and sperm chromatin compaction. The body is first seen the body shows some forward motility in the body of the epididymis, and then in the tail there is normal fertility, the sperms are fertile, there is a distal droplet, and there is high disulfide linkage. Transport takes about 9 to 13 days depending on the species. The duration of spermatogenesis, entire spermatogenesis takes about 50 to 60 days in boars, 60 to 70 days in bull and ram. The daily sperm production would depend on the age of the bull, breed and many other factors and can range from 3 billion to 6 billion sperms per day. Now we talk about the spermatogenic cycle and wave. A spermatogenic cycle is defined as the time it takes for the reappearance of the same stage of spermatogenesis within a given segment of the tubule, observed in the cross sections. Each stage of the cycle follows in an orderly sequence along the length of the seminiferous tubule. A spermatogenic wave is a sequence of segments showing the complete series of cell associations corresponding to the stage of the cycle of the seminiferous epithelium. Or in other words, the distance between the same stage is called the spermatogenic wave. One tubule can contain numerous complete waves. Adjacent segments of the tubule evidently communicate in some unknown manner. These cycles of spermatogenesis are essential for continual sperm production. We can say the wave is to space what the cycle is to time. The duration of spermatogenesis varies, and the number of stages within a spermatogenic cycle and the number of cycles required for completion of spermatogenesis varies between species. There are different stages of the cycle in the bull of about 14 days each. Approximately 4 cycles within a given region of the tubule occur before an A1 spermatogonia is transformed into a spermatozoa. So this is the duration of spermatogenesis in different species: bull, ram, boar, stallion, and humans. The consistency of these cellular associations results from two phenomena. At one point of the tubule, new spermatogonia begin their divisions and engage in spermatogenesis at time intervals of constant duration. Once the germinal cells are involved in spermatogenesis, their rate of differentiation is always the same, and each step has a fixed and constant duration. So this is again to show the spermatogenesis complete in the stallion, which takes around 57 days. Again a picture showing the spermatogonia transforming into a new group of cells that initiate the cycle. Then there's formation of the primary, secondary spermatocytes, and then the spermatids and the spermatozoa. These are the round spermatids which are then transformed into fully formed mature spermatozoa. So every 13.5 days sperm are released from this point. Then there is a coordination of stages which forms the waves. Some portion of the seminiferous tubules is always releasing sperm. Then there is compartmentalization. The mitosis, spermatogonia A1B primary spermatocytes occur in the basal compartment, and meiosis, the formation of primary spermatocytes, secondary spermatocytes, and spermatids, and spermiogenesis occurs in the adluminal area, whereas the spermiation occurs in the lumen. Then about the blood testis barrier, Sertoli cells and associated tight junctions form the blood testis barrier. They first appear at puberty. They are induced by FSH, and after puberty they can be maintained by FSH and or testosterone. They are essential for the meiosis. Then the seminiferous and the interstitial tissue are shown in this picture. Then the sperm transport, we have already talked about it. From the seminiferous tubules, the sperm reach the retitestes to the caput epididymis, the corpus, and the chorda, which is the reservoir for the sperm. And then they are released to the vas deferens, from where they are ejaculated at mating, along with the secretions of the accessory sex glands to form what we call the semen. The fate of ejaculated spermatozoa. The production of spermatozoa does not cease in sexually inactive males. The two most likely ways of disposal of surplus spermatozoa are that either they are voided in the urine or that they are reabsorbed in the epididymis or vas deferens. It has been hypothesized that in the normal androgen maintained epididymis, degradative pathways are present in the luminal fluid that are constitutively inhibited by survival signals emanating from the epithelium. Then the hormonal regulation of the spermatogenesis. The hypothalamus affects the anterior pituitary by way of GnRH, and this causes the release of FSH, which has effect on the Sertoli cells, which facilitates spermatogenesis. The LH, which has effect on the Leydig cells, leading to release of testosterone, and the Sertoli cells also release inhibin. The estradiol and dihydrotestosterone are involved in the spermatogenic process, and the FSH controls the secretory activity of the Sertoli cells and is responsible for the synthesis of androgen binding protein that bind the androgen protein and stabilizes the concentration of testosterone. The testosterone and inhibin inhibit the secretion of GnRH by the hypothalamus and LH and FSH by the pituitary. The Sertoli cells have receptors for follicle stimulating hormone and testosterone, which are the main hormonal regulators of spermatogenesis. Hormones such as testosterone, FSH, and luteinizing hormone are known to influence the germ cell fate. Their removal induces germ cell apoptosis. The Sertoli cells, they provide nutrition and support to the developing germ cells. They release spermatids as spermatozoa. They phagocytose degenerating germ cells and the residual bodies. They secrete the androgen binding protein, calmodulin, plasminogen activator, inhibin, and they act as the blood testis barrier. Sertoli cells convert testosterone to dihydrotestosterone. The spermatocytogenesis is completely arrested at the primary spermatocyte stage in a hypophysetemized animal. So meiosis 2 is hormonally independent, but meiosis 1 is hormonally dependent. The factors affecting spermatogenesis include the physical factors, irradiation, hyperthermia, photoperiod in the seasonal breeding species, and then the chemical factors, cadmium salts, alkylating agents, diamines, hydroxyurea, DDT, diethylstilbestrol, environmental pollutants, etc. They negatively affect the spermatogenesis. The nutritional factors, low or high plane of nutrition, vitamin A deficiency also affect the spermatogenesis. Then exogenous administration of steroids or stress also affect the spermatogenesis. The genetic factors, hereditary conditions such as testicular hyperplasia, free Martin, inbreeding, and hybridization. And pathologic factors such as cryptorchid, testicular degeneration, fibrosis, scrotal or inguinal hernia. The age of the bull has a direct effect on the spermatogenesis. Bulls can remain in service until they are 10 or 12 years of age. However, this is not the norm and usually remain in the herd for 4 to 5 years. So let us hurriedly recapitulate what we have learned. Spermatogenesis involves three phases, namely spermatocytogenesis, meiosis, and spermiogenesis. Spermatocytogenesis involves activation of spermatogonia and mitotic division to yield 16 spermatocytes. The primary spermatocytes undergo two meiotic division to yield 64 spermatids. In spermiogenesis, the spermatids undergo morphogenic changes without further division to form the spermatozoa. Spermiogenesis involves four phases: the Golgi phase, cap phase, acrosomal phase, and maturation phase, during which there is nuclear condensation, acrosomal and flagellar formation, and cytoplasmic reduction. Spermiation is the release of mature sperm from seminiferous tubules into the lumen. The spermatozoa mature in the epididymis and stored in the tail of the epididymis. Factors that affect spermatogenesis include age, nutrition, genetics, and pathological factors. You can contact me at my email gnpobs@gmail.com. This is my LinkedIn link and this is my YouTube channel link. So kindly listen to the lecture completely, give your comments, likes, and share and subscribe the video if you like it. Thank you so much.